Question number 1. More than 50% of mass of chromatin is DNA, protein, RNA, fiber. Correct answer is option B, protein. Question number 2. Chromosomes greatly alter their level of compactness as cells progress through the cell cycle, varying between highly condensed chromosome metaphase just before cell division, interphase, nuclear matrix, chromosome loops. Answer is option A, metaphase. Question number 3. The end of the linear chromosomal DNA are protected from degradation and gradual shortening by endonuclease, exonuclease, telomere, cap structure. Correct answer is option C, telomere. Question number 4. Dash is the more diffuse region of interphase chromosome consisting of inactive region in the 30 nanometer fiber from heterochromatin, chromatosome, nucleosome, euchromatin. Correct answer is option D, euchromatin. Question number 5. 5CG3 CpG sequence in mammalian DNA are normally methylated on guanine base, cytosine base, thymine base. All of above are false. Correct answer is option B, cytosine base. Question number 6. Which of the following is true for satellite DNA? Occurs mostly near the centromere of chromosome. May be involved in attachment of micromitotic spindle. Consist of huge number of tandem repeats of sort up to 30 base pair sequence. All of above are correct. Correct answer is option D. All of above are correct. Question number 7. The coding region of gene are interrupted by sequence: intron, exon, chromosomal loops, stop codon. Correct answer is option A, intron. Question number 8. When the some region or locus of chromosome has two or more slightly different DNA sequence in different chromosome or individual of the same species, these are described as satellite DNA, dispersed repetitive DNA, polymorphs, moderately repetitive DNA. Correct answer is option C, polymorphs. Question number 9. SNP can occur within the short sequence that are recognized by restriction enzyme. Thus, the length of fragment generated by cutting a DNA molecule with restriction enzyme could be different for each allele. This is known as polymerase chain reaction PCR, restriction fragment length polymorphism RFLP, real-time PCR, variable number tandem repeats VNTR. Correct answer is option B, restriction fragment length polymorphism. Question number 10. In the retrovirus, the single-strand RNA molecule is converted to a double-stranded DNA copy TS replaces U, which is then inserted into the genome of the host cell. This process has been termed transcription, translation, reverse transcription, central dogma. Correct answer is option C, reverse transcription. Question number 11. For pure DNA, the value is 1.8. The value below 1.8 suggests protein contamination, RNA contamination, lipid contamination, primer dimer. Correct answer is option A, protein contamination. Question number 12. The thermal denaturation of DNA may be reversed by cooling the solution. Rapid cooling allows the formation of local region of double-stranded DNA formed by the base pairing of short region of complementary within or between DNA strand. The only complementary DNA strand to find each other. Completely double helix formation. All of above are correct. Answer is option A. Question number 30. A bacterial enzyme uses the energy of ATP hydrolyzed to introduce negative supercoiling into DNA hence removing positive supercoiling generated during replication. Nuclease, DNA polymerase, helicase, DNA gyrase. Answer is option D, DNA gyrase. Question number 40. The major protein component of chromatin referred as histone, histone-like protein, RNA polymerase protein, transcriptional factor proteins. Answer is option A, histone. Question number 50. In some organisms, examples are known where the sequence of the messenger RNA is altered after the transcript from the DNA in a process known as RNA editing, RNA splicing, methylation, phosphorylation. Answer is option A, RNA editing. Question number 60. In prokaryotes, the enzyme transcribes a gene from the promoter, DNA polymerase, primase, RNA polymerase, DNA ligase. Answer is option C, RNA polymerase. Question number 70. The coding region of the messenger RNA are translated into protein by RNA polymerase, ribosome, reverse transcriptase, polycistronic. Answer is option B, ribosome. In eukaryotes, question number 80. In eukaryotes, transcription takes place in the nucleus, translation expressed in cytoplasm. The messenger RNA are consequently longer lived. All of the above are correct. Answer is option D. All are the correct answer. Question number 90. In eukaryotes, the RNAs are modified in the nucleus by capping at the end of 5' end, the addition of poly A tail at the 3' end, the addition of cap structure at the 3' end. A and B are correct. Answer is option D. A and B are correct. Capping at 5' end, addition of poly A tail at the 3' end. Question number 20. The introns in pre-mRNA must be removed to produce continuous protein coding sequence in a process known as methylation, splicing, capping, post-translation event. Correct answer is option B, splicing.